My name is Anissa Mitchell. I'm with PMD Alliance. I'm glad to have you guys joining us today for our program. We're going to be talking about telling Parkinson's disease from Parkinsonisms with MRI-free water analysis. And if you're like me, you're like, what? So we're going to get to hear something cutting edge. If you are familiar at all with the term Parkinsonism, Parkinson's disease is one of those. And sometimes it can be difficult to figure out what it is somebody has because the symptoms can be overlapping or similar. And so having tools that can help us differentiate is so very important. So we're excited to learn about some new diagnostics and ways that we can maybe help to be more uh, definitive about a diagnosis. So with us today is Dr. David Vaillancourt. He is the chair and professor in the Department of Applied Physiology and Kinesiology at the University of Florida. His research focuses on how the brain regulates voluntary and involuntary movement with specific focus on motor disorders. His research program uses advanced neuroimaging techniques to study the functional and structural changes in the brain of humans and animal models that span Parkinson's disease, tremor, ataxia, and dystonia. And he has conducted studies investigating interventions, including rehabilitation, surgical and pharmacological interventions, and published work in many, many journals. So he's well established, and we're very excited about having him. So I'm going to hand this over to you, Dr. Vaillancourt.
Sounds good. Thank you, Anissa. Thank you for having me, and thank you all for taking time and coming into this Zoom environment. So I can see everybody's face, at least most of you right here. So if during the session here, if you have a question and you just want to ask it right then, that's perfectly fine with me. I'll let Anissa sort of drive the questions. But anytime you want to ask a question, I'm totally okay with it. So can everybody see the screen okay? Yep, good. So I'm from the University of Florida. This is Gainesville, Florida. Some of the nice buildings and attractions in Gainesville, Florida. Spent my time in PhD at Penn State University. I'm not a medical doctor, I'm a scientist. And I do research mainly focused on Parkinson's disease, but also other movement disorders. So we also study dystonia, ataxia, Alzheimer's disease, a number of other conditions as well. So I've been a scientist for, several decades now, and really got interested in the area of Parkinson's disease because I was primarily interested in the study of movement, and that's how I got into studying Parkinson's. But like many of you, I'm also a caregiver. So my parents recently moved in with us. We have 3 daughters. One is in college, one just graduated from high school, and the other was a freshman in high school. But my parents, my dad has Alzheimer's disease. He's on the left, and my mom is 82. She has MCI right now, but she's doing really well. She's doing great. But they moved in with us, and so it's a big happy family. And so I'm also a caregiver, so I definitely can recognize the challenges of people who take care of loved ones. And I want to say hats off to certainly caregivers who are on this, and appreciate everything you guys do. So this video, you may have seen it before. It's not mine, but I've gotten it from others. It's the oldest known video of a patient with Parkinson's disease. You see, this is certainly the pre-Cinemat, pre-levodopa era. Okay, so you'll see many sort of very prominent features of motor symptoms in this person. So difficulty turning, difficulty walking, tremors in the upper limbs and also the lower limbs. You see a stooped posture in the person. You'll see rigidity in the person. And these are the signs that a physician uses now to still diagnose Parkinson's. So they're still looking for tremors. They're still looking for problems initiating movement. They're looking at the speed of the movement, and they're also looking at rigidity. And many of these features are still used today. But as we start thinking about some of the big problems in the field of Parkinson's disease, one of the big problems is we know that every year there's at least 90,000 newly diagnosed Americans who are diagnosed with the disorder. And so we also know that worldwide, there's roughly about 10 million people diagnosed currently with the disorder. This number was recently updated, and now the estimates suggest it's about 10 million. We also know that in the next 25 years, the number of people diagnosed is meant to double. So that number, 10 million, is going to go to 20 million in the next 25 years. So that kind of just shows you that the problem is growing, and it's not that we're all drinking the wrong water, or we live in the wrong city. A lot of it is patient care and access. So a lot of the growth is actually in China. So like the western parts of China, which are becoming more accessible to healthcare over the next several decades, that's where a lot of the growth is. There is a lot of the growth that's also in North America, but most of it is actually outside of the United States in terms of the real growth. But Parkinson's disease is faster growing than Alzheimer's disease currently. So many of you are certainly aware of dopamine medications. That has been, I think, one of the biggest innovations in research and treatment for people who have Parkinson's disease in the last several decades, or last 50 years or so. It's just been a tremendous benefit to patients. If you had some of these medications that work for Parkinson's in the Alzheimer's disease field, it would be way better for patients with Alzheimer's disease. So even though the medications are not perfect, they do help in a number of ways. But even still, the dopaminergic medications are really treating symptoms, as many of you know. So it's like you have a headache, you take a Tylenol, but you have to take another Tylenol like 4 hours or 5 hours later because that headache is coming back or your fever is coming back. Just like that same thing, the dopamine medications are not affecting the disease process, they're basically treating symptoms. Well, in the Parkinson's disease field, there's actually 0 medications approved for slowing down the disease process. Okay, so that's kind of the sobering news, is we still today have no medications which are slowing down the actual progression of the disease. Nothing approved by the FDA that slows down progression. However, in multiple sclerosis in the MS field, you have well over 14 medications, last time I checked, that are slowing down disease modification or disease progression in multiple sclerosis. And you might ask yourself, why? Why was MS, why was that? Well, early in the early 1980s, when MRI manufacturers started making MRI machines, General Electric, Philips, Siemens, these big manufacturers, people started to recognize the lesions in the brain of MS patients. And these lesions, they can count them. You can see them on one of the most widely available MRI images, which is called a T1 weighted image. So if you were to use Google and search for MRI, you'd probably see a T1 weighted image quite quickly. But the point is, in the MS field, they could see these lesions, they can quantify them, you can count them, and these were used in outcome measures for clinical trials. And so they helped the MS field recognize and test medications in a really proactive way. In the Parkinson's field, MRI has not really been that useful clinically. And if you've had an MRI, most of the time they're trying to rule something else out. Well, in the Parkinson's field, one of the biggest problems is the misdiagnoses. Some of you probably have had your own journey. We could sit here and talk about that journey. I'm not going to ask people this question right now, but I'm sure each of you has had a different journey in how you came to a diagnosis if you're diagnosed with Parkinson's or your loved one is diagnosed. And many of you will probably have experienced time that it took to get a diagnosis. Some of you may have had physicians tell you the wrong diagnosis, and then it subsequently gets to the right diagnosis. I bet that journey's happened. But in clinical trials today, where new medications are being tested, roughly 5 to 15% of patients in clinical trials don't actually have Parkinson's disease. They have something else. And so if you think about that, so if you think about a trial of, say, 300 people, that's up to 45 people who might have a different diagnosis. That's a lot. And the type of outcome that can really throw off the outcome of a clinical trial testing patients in that outcome study. So it's like Swigert and Lovell said from the space station in 1970, they said, Houston, we have a problem. And the problem that I would suggest is that there are a lot of misdiagnoses clinically in the Parkinson's field. And these misdiagnoses also perpetuate clinical trials that are testing new medications. And that is one of the reasons I think that a lot of medications are not working, is because they're being tested on people who don't actually have Parkinson's disease. So this is current standard of care, which I'm sure many in the Zoom room here have experienced. So, patient goes and sees doctor. Doctor may order an MRI, they may not. If they order the MRI, what they're doing nowadays is ruling something else out. So they're basically saying you don't have a stroke, or you don't have a vascular Parkinson's, or you don't have a tumor or something like that. They're not using it proactively in diagnosis. And then the patient visits with the doctor, or the caregiver and the patient visit with the doctor, and then they get a diagnosis. In the field, if you look at the gold standard for diagnosis, would be after somebody has passed away, what's called postmortem. And if you look at the postmortem diagnosis and you compare that to an early diagnosis of Parkinson's disease, and you look at the literature of the published studies, they range from 55 to 75% accurate. So that means in the first 5 years, a Parkinson's disease diagnosis is only 55 to 75% accurate. Now that is a fairly low number, and it's probably lower than most people recognize. So you might ask yourself, well, why does it matter? Why does getting it right matter? Well, there are basically different medications that would be prescribed for somebody who has Parkinson's disease versus other forms of Parkinsonism. So other forms of Parkinsonism would be what is called multiple system atrophy, or progressive supranuclear palsy, or there are other forms of Parkinsonism as well, like vascular Parkinson's, cortical basal degeneration. These are other forms as well. The most frequent ones would be either multiple system atrophy or progressive supranuclear palsy. Deep brain stimulation, some in the room may have DBS, some may not, but roughly 10% of patients with PD, 8-10% have had, might consider going for DBS. But DBS is only approved by the FDA for Parkinson's disease. It's not meant for somebody who has multiple system atrophy. In the brain bank here at the University of Florida, there's certainly a good number of people who've had deep brain stimulation, but don't have Parkinson's disease. Okay. The prognosis is different. So patients with Parkinson's disease tend to live longer than patients with these other diagnoses. And then also a big one is the development of new medications. These trials that are ongoing typically are having a good portion of individuals who don't have Parkinson's disease. So the statistical power you need to overcome that is causing the trials to be larger, it's causing them to be more expensive, and that itself also is slowing down clinical trials. So I'm not going to get too technical, but I want you to at least know what we're doing. So basically we're measuring what is called the diffusion of water within the brain. So if you take a water molecule and it diffuses, if there's no constraints on that, then it's going to diffuse randomly. Okay, it's going to diffuse all over. If you have constraints, such as like something in the brain, imagine you have like a tube in the brain, or you have something, even a bowl of water would provide constraints. And that diffusion is going to go along a certain orientation or a certain vector. And so in that specific type of process, you're basically measuring how the constraints on that diffusion process occurs. So in the brain, as cells die or as tissue changes, or the environment around cells change, then that orientation of the way water is moving also is changing. And so the constraints on the diffusion of water molecules is going to differ depending on the level of changes in the brain of an individual as they progress. At least that's the hypothesis that we have. So if you take this as it's called an MRI voxel. This is what this square would be. And so these little circles are cells in the brain, and they're going to have diffusion going on inside the cells, which is the red, and the blue is the what we call free water, and that's basically how much free water is occurring outside the cells. And so what we're basically measuring with free water from a standard sequence on a standard MRI machine is we're trying to see how much water content is in every voxel in the brain. And the more water that's there, we think is basically indicating less tissue and therefore more degeneration. And that's going to basically be a marker that is really important in Parkinson's disease. That's the idea. Okay, so what makes us think that? So early on, this is about 10 years ago, we published a paper, and and it was basically following a group of people with Parkinson's disease who had been diagnosed only for a couple of years, and we followed them for a year, basically 12 months. So this image right here, let's see if I can put a laser pointer. Okay, so they got this little laser pointer. So this little dot right here in blue is showing you high free water on the left side of the midbrain, and this is showing you high free water on the right side of the midbrain. And the same patient right here is a year later, and you can see that blue is basically worsened, okay? And so as the blue signal gets brighter and brighter blue, that indicates higher free water. Now, this part of the brain is called the substantia nigra, this little blue area, and that's where dopamine is produced. So many times when you're taking an individual takes dopaminergic medication, it's trying to alter the way that this part of the brain is functioning. So we're measuring the content of free water within that part of the brain. And what we found was, one, the group in black here, the Parkinson's individuals have a higher free water in the substantia nigra at baseline, and then it gets worse over a year. So it goes up, whereas the healthy group didn't change over a year. So what that means is we're able to actually track the changes in the substantia nigra over a 12-month period of time. So we're actually following the disease process over time. And this becomes useful because then we can use that measure as a readout in a medication trial or a new drug trial, seeing if we can test a new drug. So everybody's heard of Michael J. Fox. So the Michael J. Fox Foundation has a large international cohort study called the Parkinson's Progressive Marker Initiative Study. We actually downloaded that data, and then we asked the same question that I just showed you in the slide before. We basically asked the question, are people with PD progressing in the substantia nigra? Is their free water changing? And this is showing you the change on this y-axis here, right here. And it's basically showing the PD group in blue is worsening over a year. And that basically replicates the study that I showed you before. So we're basically finding the exact same finding. In addition, we followed this group, which the Michael J. Fox Foundation did, for 4 years. And you can see free water in that substantia nigra region is going up year after year after year. And what that taught us in this study was that, one, we're seeing this across a Gainesville community cohort, but we're also seeing it in an international cohort. And that strongly suggests that this is a real signal. It's something that's reliably being measured. And also in the Michael J. Fox Foundation cohort, it's also being measured across many different MRI scanners. And in any clinical trial that would be performed on a new medication, you're going to have a lot of different scanners involved. And that's not a trivial thing to measure something across MRI scanners, because you're getting something quantitative, and that's a difficult thing to measure in an MRI world. And so the fact that we're able to measure this in this cohort suggests that it can be used in clinical trials in the future. So we ran a clinical trial in like late 2017, 2018, 2019. The paper came out in 2021. And what we did was we tested a medication called Rasagiline or Azilect at 1 milligram a day. And we did this because there were some suggestions in the literature. There was a big paper in New England Journal of Medicine in like 2013, which suggested that 1 milligram of Rasagiline might be disease modifying. So we ran a clinical trial, and we used free water imaging as an outcome measure. And what we found was while Rasagiline can still improve symptoms, so it's FDA approved to improve symptoms of the disease, we found no evidence that it's slowing down the disease process. So that suggests that it's probably not a disease-modifying medication, or at least we didn't find any evidence for that. So I'm going to also kind of transition to something you may not have heard of. You also may have heard of it. There's something called REM behavior disorder, which is rapid eye movement behavior disorder. It's a sleep disorder. And what happens is people with REM sleep disorder, 50% of these individuals go on to get Parkinson's disease. So it's a high risk to subsequently get Parkinson's disease in the future. So the REM behavior disorder individuals are in a very interesting cohort to study, because you're basically studying this cohort before they ever get a diagnosis of Parkinson's disease. So what this is another group, a group out of China, which used this same technique of free water imaging. And what they found was in the substantia nigra, they found the group of people who have REM behavior disorder have higher free water in the substantia nigra, higher than a healthy control group, and then also a little bit lower than the people who have Parkinson's disease. So it kind of suggests that basically this measurement of changes in the substantia nigra is occurring before the diagnosis of Parkinson's disease. Okay, so at that point in this journey that I'm telling you about, we learned a few things. One, we can measure free water in the substantia nigra. We've learned that we can measure it over time, and we've learned that it's being reliably measured in REM sleep disorder patients before they get Parkinson's disease. Okay, so that suggests that this signal is real, it's reliable over time, and it suggests that we can measure it really early on. So we then started to put together a data set from across the world, because what we wanted to then develop was basically an automated tool that might be able to help in diagnosis. And so we wanted to also look outside of the substantia nigra, because the substantia nigra is one part of the brain, but many parts of the brain might teach us about different forms of Parkinsonisms and differentiating them from Parkinson's disease. So we had a group of healthy controls: Parkinson's disease, multiple system atrophy, and progressive supranuclear palsy. There were 17 different MRI scanners in this study, over 1,000 patient studies or cases, and 8 different international cohort sites. I'm not going to go through the next slide in a lot of detail, but the key finding was that we were able to, with a high level of accuracy, differentiate Parkinson's disease from multiple system atrophy and progressive supranuclear palsy. In addition, we were able to differentiate multiple system atrophy from progressive supranuclear palsy based upon a clinical diagnosis. Now, this was what one calls a retrospective data set. So we're using data that's already been collected and asking these questions, and we're able to show that indeed we were able to do this using what is basically a machine learning procedure. And similar to like your phone. So I have a phone that, this is my daughter, this is my youngest daughter, but on my phone, I have a really old phone that when I touch it, it unlocks it. Okay, so basically it's looking at my fingerprint and looking at the key features of my fingerprint, which is hopefully different from everybody else's fingerprint, so nobody else can unlock my phone. Well, in this basically procedure, we're basically looking at different patterns in the brain. And we're basically having a machine learning procedure that's looking at those patterns and then determining if somebody has Parkinsonism or somebody has Parkinson's disease, similar to the way you unlock a phone in that case. It's basically determining yes or no, you have Parkinson's disease or you have Parkinsonism. And we're using that sort of machine learning procedure to, in an automated way, to improve diagnosis in this particular study. Okay, so this was about 2019, 2020. And on this journey, then we basically realized that we might have something really interesting in terms of improving diagnosis for Parkinson's and Parkinsonism. So we started to work with the Parkinson Study Group, which is an it's really international, but it's really North American. So US and Canada neurologists who run clinical trials for Parkinson's. Really good group of individuals. And we were able and very fortunate to work with them. And they helped us design the study. And we also were able to get it funded by the National Institutes of Health. And so what we wanted to do was prospectively test the algorithm that we developed previously and determine if we could prospectively diagnose patients in this new study. So in this study, we were trying to improve diagnostic accuracy in a clinically diagnosed cohort, and also we have a large group of pathologically confirmed cases. We wanted to also develop an automated tool that could be used by a neurologist or a radiologist in the future. We also think the tool could be helpful for inclusion into clinical drug trials for new studies that are starting. We also wanted to use a technology that's clinically available. So we didn't want to develop something new that couldn't be implemented. We wanted to develop something that can be implemented on MRI scanners currently available. And in doing so, we also wanted to have a safe and reliable data transfer tool, because hospitals and radiology and imaging centers are going to need something that's safe and secure through that process. So we ran a study across 19 sites in the United States and 2 in Canada. We had 21, of course, total sites, And you can see many of the sites here in this image. One of the unique parts about the study is we had 3 blinded neurologists who were assessing the diagnostic accuracy of the patients. So we were able to get, I think, the best gold standard clinically that would be available in assessing the algorithm's performance. We also use different scanner manufacturers. So, like the elephant in the room in the MRI world is there's different manufacturers of the MRI scanners, and they all compete with each other because they're trying to sell their MRI machines. They don't want to talk to each other. They're actually incentivized not to talk to each other because they make more money when they don't talk to each other. So a hard thing to do is to develop software that works across them. And so that's the real key here, is that we've developed software that works across the major manufacturers for MRI machines worldwide. So Siemens is German, GE is the United States, and Philips is the Dutch. So we also have a group of autopsy confirmed imaging here as well. So we're able to basically assess our algorithm's performance in 49 cases who have autopsy confirmation. So I'm not going to go into the details of the scanning, but what I want to tell you is that these scanning sequences can be performed on just about every 3T MRI machine, certainly in the United States. So, if a scanner's been developed in the last 10 years or bought in the last 10 years, there's going to be a sequence that's just already FDA approved, which would be able to be implemented, that would basically be able to use our software. Okay, so this is like the primary outcome of the study, and it's basically assessing the performance of the prediction algorithm on different scenarios. So basically, this is the Parkinson's disease versus atypical Parkinsonism, and this is a fancy statistic to tell you that it's working really well. It means that with 96% area under the curve, which is basically telling you the level of accuracy that's going on in differentiating Parkinson's disease from atypical form. This is multiple system atrophy from progressive supranuclear palsy, Parkinson's disease from multiple system atrophy, and Parkinson's disease from progressive supranuclear palsy. All the models did really well in this study. Now, MRI is like an infinite resource, okay? So I showed you here is this one model, but then we ran the experiment 49 more times, okay? And we just basically ran it again and again and again and again. And we did that because as a scientist, I'm very cautious. I don't like saying things unless I actually can demonstrate them. And so when we ran the experiment over and over again, we wanted to ask the question, do we get a similar result? Because if we don't get a similar result, that means that what we did initially was likely due to chance and not reliable. So when we ran it 49 more times, we got a very similar result. That was the good news. So basically, all these orange lines are different simulation runs where we basically got a similar result. So the other key part about this study is we tested the algorithm that we had developed on a separate group of people who had donated their brain and had an autopsy performed and had an autopsy confirmation of which diagnosis they had. So the imaging scan occurred 4 to 94 months before autopsy occurred. And we were able to predict 46 out of 49 pathologically confirmed cases. Amazingly, these patients had been followed up until death, and only 81% clinical diagnostic accuracy. So even though the physicians had been following them for years, they still were only 81% accurate in predicting the true diagnostic accuracy. So our imaging algorithm still had an over 12% gain in diagnostic accuracy compared to the final clinical diagnosis. Okay, so this is something my wife put on my computer screen. I actually am sitting at the same computer screen now, so the note is still on my computer screen. This is during COVID when we all had to go home and work from home for a period of time. And she put this actually on my computer, but also our daughter's computers as well, telling us to focus because it can be easy to get distracted while working from home. So it's just basically kind of meant to kind of give you a transition to say, well, what was next? What do we do next? And so during this process, one of the things I realized was if people are going to use this software, then it has to become commercially available. So I could put the software for free on the internet for people to use, and nobody would use it because the way hospital systems, that's not the way our hospital systems work. That's not the way healthcare works. Basically, they're only going to use stuff that's approved, that's basically commercially viable, and that they can actually utilize and that they trust. So we started a company in 2021, myself and a computer programmer named Angelos Bonpudas called NeuroPax. Neuro, of course, is the abbreviation for neurology, and PACS is a radiology term called PACS, so Picture Archiving Computing Systems. And all radiologists are using software, which is called a PACS environment or PACS software to view MRIs. So we've had a lot of successes in the company so far over time, and currently have submitted to the FDA, the United States FDA, for approval of the software to be used clinically. And we're in that process right now, and hopefully this will be approved in the near future. So this is just kind of a demonstration I'll show you of the software being used in real time. So this is just an interface. The software actually sits in the cloud on like an Amazon cloud. So there's somebody dropping an image in, and it's basically de-identifying the data. So that basically strips any identifiers from the image file. And then you're going to see a couple of different results. These are just early prototypes of the software showing you how it can be used. So this person had a predicted MSA diagnosis, you can see. And so they would have had an atypical Parkinsonism and then an MSA diagnosis. This person had a PD diagnosis. And then the next one would be an example of a PS. Sorry, this one has MSA also. So the software sits within a cloud environment. So the MRI would be taken. It's then transmitted to a cloud-based server. We use basically an Amazon, just like you order from Amazon. They also have wonderful computing power to process data in the Amazon cloud. Machine learning occurs, the classification occurs, and that we basically produce a diagnostic report within about 2 hours of time. The software is set up to handle like 500 at this point, 500 cases every 2 hours. So, we can scale it upward very easily. So I just kind of want to summarize, and then I just wanted to take questions from anybody that had questions. So the free water, I've shown you some evidence that it's sensitive to changes in the substantia nigra in early stages of PD, and also in patients who have REM behavior disorder. I've also shown you that an automated form of the software called Automated Imaging Differentiation of Parkinsonism, AIDP, is detecting atypical Parkinsonism accurately in a multi-site cohort. I've shown you the AIDP works across different MRI machines, so Siemens, GE, and Philips. The AIDP is also in agreement with neuropathology from an autopsy confirmation, up to 93% accurate. And also the AIDP is in progress as a software tool. And once FDA approvals occurred, then it will be available to be used clinically. I want to thank you all very much for your time and see if I can take any questions for you. This is a picture from Gainesville of the University of Florida.
Thank you. It is a beautiful campus. Okay, so I'll start off first how important something like this is for people. One comment that came early on in your presentation was that this person was diagnosed with diseases incorrectly before meeting with their movement disorder specialist and getting a proper Parkinson's diagnosis.
Yeah, so That is not an uncommon story where you might go see your general neurologist or maybe even primary care physician, and uh, the diagnosis can be wrong for a while, and then you see a movement disorders physician, and then that become more accurate. But even movement disorders physicians make errors. They do make less errors, I would say, but they certainly still make errors. In the cohort that I showed you, where we predicted 46 to 49 brains, but the clinical diagnosis was only 81% accurate, that 81% was still from movement disorders trained physicians.
So once this becomes available, do you see this even being used to assure if someone has already been given a diagnosis with Parkinson's, maybe they've been diagnosed for a few years, maybe but they're contemplating DBS, and I know you said obviously we don't want to implant somebody who isn't Parkinson's. Is this something that would be potentially utilized to assure that we would be implanting in the right person?
Yes, absolutely. I think it's a very good way it can be used clinically, is to assure that um, that a person who's being considered for DBS actually has a Parkinson's disease diagnosis, doesn't have an atypical form. I think it would be certainly the efficacy you would think would be better for those people who have a true diagnosis of Parkinson's disease rather than an atypical form. I also think insurance companies might be interested in not wanting to pay for individuals who don't have Parkinson's disease to get the surgery.
100%. There was a question whether this is a regular MRI that everyone has easy access to, an advanced model.
Yeah, so in general, it's a pretty regular MRI to get access to. These types of MRIs are performed regularly clinically now. They're just not always used in Parkinson's disease. So many times a diffusion MRI is actually standard for like workup on a person who's had a stroke or a suspected stroke. It would also sometimes be used in traumatic brain injury, like a person who had a concussion. In the MS multiple sclerosis, it also can be used there. So it's not necessarily used in the Parkinson's field that much from radiology, but the type of sequence is sitting on scanners. It's easy to perform. It takes about 6 to 10 minutes, depending on the scanner and the software.
It's amazing. So I guess this question is relevant then. So could this software and MRI be modified to be used to differentiate between Alzheimer's and Lewy body dementia and frontotemporal dementias?
Yes, it can. It's a really good question. Very smart question. And we are working on that right now. So yeah, we actually about to submit a manuscript on that. The data look just as good. So
that's incredible. It's very exciting to see that this is becoming a potential tool for helping us to determine what exactly is going on. And I guess that goes along with my next question. So would you consider this then a standalone diagnostic tool, or would you pair this with something else such as the skin biopsy?
yeah, I think in the Parkinson's world and the Alzheimer's world, ultimately diagnoses will come down. No matter what test you have, it'll come down to a physician. I think that's number one. Number two is in both the Parkinson's world and in the Alzheimer's world, I think you're going to have a skin biopsy. Maybe in the Parkinson's, I ultimately think you'll have blood tests as well. Ultimately, in the Alzheimer's, the FDA just approved a new blood test for Alzheimer's. That was just approved in the last week or so. However, though, most of my colleagues in neurology still think MRI will be standard in the workup of individuals with both Parkinson's and suspected dementia. So either way, and also pairing this type of software with another type of biological is probably the way we think the field is going to go. And the reason is because like you can get a synuclein test from a skin biopsy, but that's really just going to tell you if you have a synuclein that's consistent with Parkinson's. It shouldn't tell you if you have like multiple system atrophy or Parkinson's disease per se. It also probably won't be able to tell you how bad your Parkinson's is. Whereas we think there's a lot of evidence that we have that the types of images that we're collecting can also tell you how bad it is. So we see it more of a combination than necessarily a competition per se, but I think it's just it's like having another tool in the toolkit for a physician.
And that is fascinating and wonderful. So if they get to a place where they actually have disease modifying treatments, is this then a way that you could potentially track whether they're working, whether it's in research or in treatment?
Yeah, for sure. So I think, the first starting point is in the clinical trials space to,
Mhm.
it's like a cohort enrichment. You could, weed out individuals who shouldn't be in the trial. That's one way to do it. Patient selection, or it could be a readout for the actual medication trial also, like to see if it's altering reward indices over time. So I think either of those are possible. Whether it's used clinically to like monitor, I doubt. MRIs themselves they're not cheap. So I kind of doubt that it would be used to monitor. Maybe you might get 2 MRIs, but to get like a bunch more, I find unlikely. But I do think it can be used in the clinical trial space to have multiple MRIs taken.
Well, and there's been a question here of how can the community help tell our doctors to use this for clinical trials, or hospitals should get it. Great technology and can help many people. So it sounds very exciting. So we would love to, our community loves to be a voice and to help. So if you have any suggestions of what we can do as a community, certainly let us know.
Okay, that sounds great. I may think of more things in the future. Now I would just say tell your friends about it, tell your family about it. I think that's, word of mouth is the most important thing. And I mentioned to Anissa that I would make you aware of the manuscript that was just recently published in JAMA Neurology. And so I would suggest going and downloading the manuscript and just taking a look at it. It's freely available. It's a PDF. So that's one thing you can do now to learn a little bit more about it, more than what I've talked to you about today.
Well, you really have helped us understand it, because I wasn't really sure what all this was going to be, and it's very exciting. Actually, it's extremely exciting news. Any idea of when you think it'll be approved for regular use?
I think it's a really good question. I think the earliest would be this year. But yeah, we're hopeful. The feedback we've received so far has been cautiously optimistic, but you never know what's going to happen. So we're in the process, and we'll be submitting back to FDA soon. So we're hopeful.
And I want to thank Andrea, who just found the journal article and dropped it in the chat. So those of
Cool.
you who are info seekers, you can grab that now or save your chat and you can go and read this. I will let you guys all know that we're not going to be able to share the slides for this, but instead you can go and read this article, because this actually, all the slides, all that data has come from that article. So but we are recorded, so you can go back and listen to it again, because there was a lot of really good stuff that I think probably repetition is the key to learning for these things. Awesome. I'll make sure we don't have any other additional questions. No, but if you do have any last minute burning questions, feel free to drop those in the chat. Also, Kelly just dropped the link where the video recording will be available in the next day or so. So if you only got part of it, if you came late, I want you to make sure you go back and listen to it, or share it with any of your friends, or even in your support group. If you're a part of a support group, it's a really good way to answer the person who was saying, what can we do to get the word out? Well, that's a great start. People self-advocating and getting the word out through your support group network. So, Dr. Vaillancourt, I want to thank you so much for your time today. And if you have any other last parting words, I'll give you that opportunity. But I really do appreciate your time and this extraordinarily exciting diagnostic tool that's coming to us.
Yeah, absolutely. I appreciate everybody's attention, and the questions are really good. So I definitely appreciate the questions and thoughts. So keep us in mind, and hopefully we'll be at a hospital near you soon.
Absolutely. It's something that we do at PMD Alliance, since we're not going to like clap as you normally would from a wonderful presentation, being hard for you to hear that, but we invite everyone to turn on their cameras if they're able and willing. And we do what was called the Great Wave of Gratitude. So we just want to say thank you so much for your time, and again, for your research, the hard work to bringing this to our future for diagnostics.
Thank you very much.
Thanks everyone for joining us, and we will see you all soon. Take care.
Bye-bye.
Bye.